Thank you. Thank you for the introduction and thank you for the invitation. It's a great pleasure to be able to share this data with you. So I've been working on a number of projects to do with COVID-19 analysis, and I'm going to share some of the data from the various projects today. So, as a preamble, as Eve has described, we have I run the Van Gies biomarker facility, and we've just moved from this picture here where we were looking predominantly at proteomic biomarkers of cardiovascular disease and a bit of oncology, to a wide range now of diseases, including diabetes, respiratory, but also looking to incorporate metabolomics and lipidomics using a variety of different techniques. I'm also director of Leicester Precision Medicine Institute, and why that's relevant is because we are very interested in deciding a type of precision medicine which is the right drug to the right patient at the right time. And underpinning this is not just the clinical cohorts, but also kind of phenomic and genomic analyses with data science and AI. And so the mass spec component is hugely important to this endeavor, really. So, as many of you know, at the start of the pandemic, there was endless challenges to do with COVID-19. Very little was known. The reliability of tests was questionable. Because of the scale of the issue, there needed to be an ability to ramp up or scale up the tests. And of course, we'd have a requirement for different tests for different settings. So in hospitals where by and large you've got acute disease, and in population settings where the vast majority of people don't have the disease. So you have different challenges all the time. I won't discuss too much about this. I guess everyone's pretty familiar now with the structure, but just to say that the two proteins we're predominantly interested in are the nucleocapsid, the NCAP, and the spike. Let me interruption. And the spike, which is so important for being able to bind to receptors and ultimately access the cell. So in the UK, March 23rd, we had the first lockdown, and I guess same as in the Netherlands, most labs shut. And that was an incredibly sad day, professionally, shocking really. So we were thinking as a translational lab, how could we stay open or get back open quite quickly? And so we'd use our skills really in developing translational assays to look for an assay that would be able to detect the virus in a variety of different samples. And this would be a kind of alternative to qPCR or RT-PCR. And there are many considerations here. So if the sample collection, this is a highly infectious disease, so it can't be used in a normal lab or can't be prepared in a normal lab. We had sample preparation, would have to be quite simple. We're pretty committed to SRM, selective reaction monitoring, for quantitative and sensitive analysis, and of course a high throughput assay. And I was thinking at the time about a 10 or 15 minute assay, but we settled down to 8 minutes and further, as I'll explain. And from these results, we'd be able to determine whether a sample was positive or negative. So we reached out to Waters in the first instance to ask if they wanted to establish a collaboration, and they actually put us in touch with Martin and Bart, who had established a international collaboration, CoV-MS, a community-based template, and they'd already set about doing a lot of the groundwork for establishing an MS assay. So I joined this collaboration. So one of the first questions we want to do if we're going to establish a diagnostic is peptide selection. And I think science has performed incredibly well over the last year. And two examples really of the speed at which people generated data and got it out, which you can see as being advantageous and disadvantageous. But so, for example, with the Ben Osborne paper, where he did a simple in silico digestion of unique peptides for the virus, I was able to establish a list of peptides by removing any peptides non-unique, so present in human or microbiome or other coronaviruses. But the limitation was that the list of peptides didn't actually fit very well with what we ultimately saw in practice. The second paper, which I've just found out this week, you spoke at the last meeting, this is an excellent bit of work. You know, April the 23rd is a month after the UK lockdown, and they described using high resolution PRM the measurement of the virus and some of the peptides which are common that we also use using, I think, nano. So the sacrifice is throughput in this particular assay, but there are 3 observations with this particular paper. And I guess the first is that the NCAP peptide represents about 88% of all signal. So the NCAP protein is the best one to target. The other observation, which I only appreciated on reading it last week, was that when they looked at sputum and fixed that in 80% acetone, it was this sample prep that was the most compatible with the subsequent proteomics workflow. And that I'll explain a bit more about that later on. And I think the end point actually was the sensitivity in equivalence to the PCR-CT values was about, if I remember, somewhere in the order of 24 to 26, which is reasonably quite sensitive. And I go on to compare that in the next subsequent slides. So Martin and Bart have been working with Leonard Martins, and he was doing some in silico work to generate a spectra profile using MS2PIP and DeepLC to get a retention time. And using these spectral libraries, Martin, who's a discovery scientist, you probably know, established 17 peptides which he felt would be useful targets in any subsequent assay. And if we look at those 17 peptides, they represent, first from the spike, we've got 2 peptides which are found in the S1 and S2 portions of the spike, which is really useful. And then we've got a pretty good coverage of the nuclear capsid. So all the red text is belong to the 17, and the blue are ones that we added subsequently as well to also have a look. And you can see that we can get some really decent chromatography with good resolution within that. So what was our role as the Van Geest biomarker facility? So we were there to collaborate and help optimize on our instruments. So we were using the Waters TQ-XS, and we'd only just bought the, or just received and had installed this instrument when lockdown occurred. Ghent supplied some of the recombinant proteins, and we optimized the transitions using a program called MassLink Skyline interface, and I'll describe that in a moment. So for those familiar with Skyline, you can set up your chromatograms, and you can get your spectra, and you can predict what you think are going to be the most dominant product ions, and from that you can generate a list of transitions which you can export. And so where MSI, MassLink Skyline interface, works is by taking the method from Skyline. It will then create a series of all the possible transitions through a series of iterative injections. It will pick 5 of the best transitions, and on those 5 transitions you will then get collision energy optimization, and then you get the final MRM method. And it works, so how it works in practice is that you put your vial in, you set the program going, and the next morning you've got your final MRM method. And we can see here in the MSI results, from the first injection to the final optimized method, we've almost got a 4-fold improvement in signal intensity for one of the peptides, just 1.25, but still an overall improvement. But this is a really useful program for getting that exquisite optimization delivered for a particular assay, for a large number of analytes, for example. So from the recombinant proteins, we were able to deduce the sensitivity in the universal transport media, which is used for holding the swab and preserving the virus. And it's really important to remember that point. And we could demonstrate good analytical characteristics with good linearity and good precision and accuracy across that. And so with this optimized method, we were able to take some patient samples from Leicester, and this is in swab on this occasion. And we got 2 peptides here, KQQ and DGI. And if we look at the peak for the standard, we find totally absent in patients who are diagnosed as negative of COVID-19 by PCR. And for patients who are positive by PCR, we can see this particular peptide present. And the same story is the same for the DGI. So now we can show with the optimized method, 8-minute runtime, we can pick up a number of peptides. I'm just showing 2 for as examples. And the results of Martin and Bart's work were ultimately that they could show linearity of their method for a wide number of different matrices, whether it was universal transport medium, VTM, viral as it's called, and then a salt preservation. And they could show pretty much a linear relationship between the mass spec response and the PCR. And then they've published this in a preprint, and it's now been accepted for full publication. Again, the sensitivity is about the equivalent of a PCR CT value of 24 to 26, pushed to 28, but it's broadly around the area. I think the key take-home message of this paper, which is a phenomenal paper in terms of data generated within it, but you can generate a high throughput MS assay that is orthogonal and complementary to PCR. I think another conclusion is that you're going to have probably the most contemporaneous of the MS equipment, so you capture that required sensitivity. I should say at the same time, we were doing some work in plasma, and I'll return back to plasma, using this method, and which was always my main intention. And we were unable, using this method, to detect the virus in the plasma. And I'll come back to that later. So then, around about the end time of that, the UK government announced the Operation Moonshot. So it turned out that this guy was interested in mass spec. And what the Operation Moonshot really wanted to do was to assess multiple tests for multiple scenarios. So they're looking at immunoassays, a whole load of nucleic acid assays, Raman spectroscopy, I think, is involved, but looking to see where they could be employed, whether at airports, hospitals, care homes, etc., etc. So this consortium was led by Purdy Barron in Manchester, has done an amazing job with this. The UK government's Department of Health have funded it, and we had a strong international steering group with, I think, Jennifer Van Eyck's on it, Martin Danen's on it as well. And we had 3 central objectives. The aim was to develop a high throughput test for SARS virus with sensitivity that was above 28, ideally. To include within that was to develop a multiplex assay for winter viruses and SARS-CoV-2, so we could differentiate if someone had the sniffles or a bit of, they felt they've got a runny nose, would it be cold or flu, or was it actually COVID? And there was also the idea of possibly including the ability to measure the antibody, and they wanted this all turned around in 8 weeks. So it's multiple centres. The way that it ran was in the first week or two, people were to try their own methods and it'd be evaluated which ones to take on. And in the end, it fell to just 4 centres to deliver phase 1, which was Leicester, UCL, London, Southampton and Manchester. And we optimised every aspect of an LCMS method. So ostensibly, we took the assay that we developed with Martin's group. We wanted to optimize the inactivation solutions, the digestion time, the injection to injection time, because everything was about high throughput and could we scale this, would it be scalable? And we also looked at software for quantification. And then we brought in, midway through, SISCAPA was made available to us. And so we were able to incorporate that into the method. So the issue with there's many issues with UTM and VTM, but one, they maintain the viruses. So to prepare them, you have to use a category 3 safety hood, which not everybody has access to. So we were very keen to have an inactivation solution from the point of sample collection. So we showed at Leicester and Manchester that 70% ethanol was sufficient to kill the virus in a reasonably small amount of time. And we're going to use this for both saliva and for swabs. So we've now got away with UTM and VTM, which the other issue is, of course, it has lots of other proteins and salts, etc. In terms of multiplex, we could demonstrate this very easily with recombinant proteins. So we had one of the great advantages of this whole process was the logistics worked very well to deliver certain reagents. So we're able to get influenza and cold virus. But one of our problems that we face with the multiplex is no one this year has had a cold or flu, right? So if you have had the snivels, it's more likely to have been COVID. But in a few of the samples where we looked for flu, we did see evidence of peptides that belong to the flu virus. So we have some evidence that we can pick this up, but we haven't been able to test it on a large enough cohort, but we're showing evidence that we can kind of do multiplex. The digestion was kind of interesting because NCAP protein doesn't have cysteines, and we could get away actually with a 15-minute digest, no IA or DDT, and we found that was perfectly acceptable, but not for spike, which requires longer tryptic digest time. But as NCAP was our primary focus, we were happy to use 15 minutes as it helped with the throughput of the assay. In terms of chromatography, so 8 minutes seemed quite good, and that would deliver 180 samples per day. But we looked up down to 1.5 minutes, which would give you nearly 1,000 samples per day. But we ultimately found that this, to some extent, gave bunching. A lot of the peptides that we're particularly interested in started to coelute, and so we settled as a consortium on a 3-minute runtime, which is still excessively, which is about 500 samples per day per machine. So the turning point for me in this project is SISCAPA technology. And for those unfamiliar with SISCAPA, it's quite smart. You take your sample, you tryptically digest it, and SISCAPA raise antibodies to target tryptic peptide. So when you have your sample, you then add your antibody that will target that peptide. You also add in at the same time a stabilized isotope label standard, and the antibody will pick up both analyte and internal standard. And by doing that, when you elute it off and analyze it on the mass spec, you're able to get a nice quantitative result. And it cleans up the sample really well. And so this is also really good because you can bring it into an automatable workflow. So we can do the extraction and digestion as a manual step, but everything else then is automatable with capture, wash, elution into the mass spec. And we can see here the chromatography now. We're just looking for 3 peptides. We're looking for AYN, ADE, and DGI. And I should, I suppose, recognize that AYN, in terms of specificity, is found in a number of coronaviruses, but ADE and DGI are specific to SARS-CoV-2. So this is, you know, a really good assay with high levels of specificity. We can demonstrate really nice with SISCAPA, really nice analytical characteristics, so linearity. We saw a great improvement in sensitivity, and we could see down to CT30 and the equivalent of CT30 in PCR. And in fact, in some of our samples, we've seen down to 30 or 32, but then it comes a question into how good is PCR at that level. And I'll talk a bit more about that in a moment. So the study that we did we were tasked with saliva, as was Manchester. And what we found is actually that we got a sensitivity. So the ability to pick up positives was about 2/3. We picked up 2/3 of all positives, but we were very good at picking the negatives. And this was disappointing. We thought we'd get better than this for saliva, and we're still working to improve that. At the same time, I thought we'd return to the plasma story and just see if SISCAPA helped with that, not thinking it would. But what was astonishing was that in patient plasma, where they're PCR positive, we could see the DGI and the AYN totally absent in PCR negative, and pre-COVID plasma we checked as well. Now, unfortunately, I can't share the rest of the data in regards to severity of disease, and I apologize for not mentioning that before, but I just can't share that at this time. But we can show that we can see the virus in plasma. So the end result of the UK government project was for saliva, as I say, 65% sensitivity, 95% selectivity. But for the swabs, which was done in Southampton, UCL, they're able to get almost 98% sensitivity, so 98% of all positives they picked up, and almost 97% of all negatives they're able to diagnose. So this is now the equivalent of what the PCR characteristics are. So we have a mass spec assay that's able to do swabs, and we're just working on the sensitivity for saliva as well, because that has great advantages in testing certain individuals. So we developed a 3-minute LCMS assay, has Winterplex capability. What the aim of the phase 1 was to bring a harmonized protocol, highly detailed protocol that would give to colleagues in hospital testing labs, and they are working to validate and get it accredited, and that's almost good. In fact, that comes to an end today, so we should have the results for next week. And I suppose one of the arching questions about variants and how that will affect things. So it seems that we've done some work looking at regional variants, and we can look at the sort of mutational frequency, and we show here that we've got in the grayed out boxes or blue boxes, these are the peptides we're selecting. The red writing with the underline is where the mutation are most frequent. And we can see, for example, with the ADE peptide, we are seeing some mutation variants occurring. But for AYN and DGI, these are seemingly free as of the moment of the mutational variant. So these are good peptides to be selecting. And we can see that in the different regions, we've got a range of different peptides that we can select that won't be susceptible to the, I guess, evolutionary pressures that are occurring. I'll finish off with one last slide, and you know, like all of us on this call, we're advocates for mass spectrometry, and it often feels that mass spectrometry is the bridesmaid in certain situations. You know, the asking to validate a mass spec protein via Western blot is a good example. But this, we asked two prestigious institutes in the UK to carry out PCR on all our samples. And what I'm showing you here is one of the centres, their evaluated CT value and the difference of that value from the other institute. And you can see that there is hardly any alignment. It is absolutely astonishing that the gold standard that we are using is PCR. Okay, so with that, I just want to acknowledge just a couple of people really particularly. Dan Lane is the PhD student who's worked, he's hardly had a day off since last May, and he has worked really, really hard. This is my team in Leicester, the COVID-MS consortium in the UK, led brilliantly by Purdy, very highly supported by Waters at Wilmslow. They've a whole team have helped us out, the Department of Health for funding, and then Martin and Bart with the original project, again highly supported by Waters and